Hello everyone. Recombinant DNA technology is an impetus to the plant genetic transformation research. Significant progress has been made in using genetic engineering methods in producing transgenic plants with the useful agronomic traits. The co-introduction of marker genes, particularly the antibiotic or herbicide resistant genes, is needed for the initial selection of the transformed plant cells. The presence of the marker genes such as antibiotic resistant gene along with the gene of desired trait in the transgenic crop is considered undesirable by many regulatory agencies. Hence, it is desirable to remove the marker genes from the transformed plants so that only the gene of interest remain in the transformed plants. In this module, we will study the various methods used to remove these markers from transformed plants. The learning objectives of this module are avoiding the use of selectable marker genes, elimination of marker genes from nuclear genome, co-transformation, site-specific recombination systems, transposon based marker excision systems, elimination of marker genes by homologous recombination, elimination of marker genes from Plastid genome. The simplest way to produce marker-free transgenics is not to use selectable markers and instead screen a large number of mutated transformants using the polymerase chain reaction using primers for the gene of interest. The second way is to use screenable marker gene in the place of selectable marker gene, which enable identification of transgenic plants in the absence of a selective agent. The use of such screenable marker does not kill the non-transformed cells. Rather, the transformed cells experience a metabolic or developmental advantage. Beta-glucuronidase can be used to cleave the glucuronide derivative of benzyladenine, thus releasing free cytokinin that stimulates regeneration of transformed cells. Phosphomannose isomerase or xylose isomerase are also used as screenable marker genes. Transformed cells expressing the bacterial genes Xyl A or PMI are able to grow on a nutrient medium supplemented with xylose or mannose as the sole carbon source, while the growth of non-transformed cells is inhibited. Genes encoding enzymes which are involved in the phytohormone metabolism such as isopentenyl transferase IPT from agrobacterium have also been used for selection of transformants. Use of role A, B, C genes as screenable markers increase the sensitivity of transgenic cells to plant hormones. Use of role A, B, C genes as screenable markers increases the sensitivity of transgenic cells to plant hormones. Co-transformation and segregation strategy to obtain marker-free transgenic plants. The selectable marker gene and gene of interest are introduced on separate tDNAs present in two different agrobacterium strains. A. On separate vectors in the same agrobacterium strain or on the same vector. The two genes can also be delivered by the direct gene transfer method such as particle bombardment. If the gene of interest and the selectable marker gene are integrated at unlinked positions, progeny plants with only the gene of interest can be obtained after sexual propagation. Removal of selectable marker genes can be achieved by site-specific recombination systems. The CRE lock system is derived from the bacteriophage P1 and consists of two components. First are two LOX P sites, each having 34 base pair inverted repeats cloned in direct orientation flanking the DNA sequence. And second, the CRE gene encoding a recombinase protein which specifically binds to the LOX P sites and excises the intervening sequence by catalyzing a crossover between the repeated LOX sites. The strategy involves the generation of plants that express the CRE gene and crossing them with plants in which the selection marker gene is introduced between two LOX sites. In the F1 generation, the selectable marker gene is excised due to the expression of CRE recombinase. The CRE gene is then segregated away in the subsequent generations. Besides CRE lock system, other site-specific recombination systems include the flippase recognition target and flippase recombinase or FRT-FLP system and the RRS systems both from yeast. chances of reinsertion of selectable marker genes are negligible. This system is primarily based on the fact that the DNA sequences located in the AC-DS repeats are excised along with the AC-DS element. Agrobacterium mediated transformation is carried out to transfer the selectable marker gene that is cloned as a part of transposable element, example AC and the gene of interest, with both genes lying within the tDNA borders. Alternatively, the AC can be introduced by crossing with another transgenic carrying AC. Transposition may result in reinsertion of the modified element along with the selectable marker gene. If reinsertion occurs in an unlinked position, marker-free progeny may be obtained after crossing and segregation. Alternatively, if no reinsertion occurs after excision of the modified transposable element, it can also result in the loss of the selectable marker gene. Homologous sequences may be the plastid promoters (PRRN) and transcription terminators present on the vector used for inserting the transgene. Recombination via the plastid promoters or the transcription terminators repeats yield two stable marker-free transplastomic DNA carrying only the gene of interest, one via recombination event R2 or the gene of interest 2 via recombination event R1. While the gene for the selectable marker AADA coding for pectinomycin resistance was removed, the promoter or terminator sequences were not repeated in the final product. A site-specific recombinase gene CRE introduced into the nucleus by transformation, which encodes a plastid targeted recombinase that excises selectable marker gene SMG from the transplastomic DNA after import into plastids. Excision of the marker genes by CRE recombinases via the target LOX-P sites as shown by the black triangles yields marker-free transplastomic DNA carrying only the gene of interest with only one recombinase recognition sequence. If two vectors are used for transforming plastids, each carrying different small regions of homology flanking the gene of interest and the selectable marker gene respectively, then homologous recombination could lead to three types of transformation events. Either the two genes could be inserted on two separate plastid genomes, or the two genes could be inserted on the same genome. Subsequently, homoplastomic population could be obtained where only the gene of interest is present on the plastid genome. The module can be hence summarized as: due to their undesirable effects, there is a need to avoid use of selectable marker genes in transformation methods, or to remove selectable marker genes after their use in selection of transformants is over, such that the transformed plants finally do not express them. Selectable markers could be avoided totally and identification of transformants could be done using PCR, but this is very tedious. Or markers that enable selection without being potentially hazardous could be used, like mannose phosphate isomerase, which enables growth on mannose medium. If transformants are generated carrying both the gene of interest and the selectable marker gene, then the latter can be removed by several methods like co-transformation and segregation, site-specific recombination or transposon based excision. Excision of markers in transplastomic plants is possible using site-specific marker excision of homology based excision. In addition, it is also possible to segregate the selectable marker carrying plastids from the ones carrying the gene of interest by obtaining homoplastomic populations of cells. The marker excision technologies are useful in overcoming the problems associated with the presence of selectable markers in the transgenic plants. Thank you.